Lovely. Thank you so much. My name is Kindly, and I work right here in Madison. For those of you traveling home today, I hope you have a safe trip. But before you go, I'm really excited to tell you about a computational method which will help analyze the type of 3D genome data that our keynote speaker just spoke about. Specifically, our framework detects higher order structural changes in the 3D genome organization using something called multitask matrix factorization. Okay, and this should all be a review. 3D genome organization, it refers to how the DNA is folded in a systematic way inside the cell's nucleus. And distant regulatory regions like enhancers and promoters can be brought together in 3D space with the help of transcription factors, triggering, which in turn can trigger gene expression and influence downstream cellular processes. And we can study the 3D genome organization with Hi-C, or high throughput chromosomal conformation technology. Hi-C measures the level of interactions or contact frequencies among genomic regions, including enhancers and promoters. And it generates a symmetric matrix, like the one you see on the right, each entry represents how often the region corresponding to the row interacts or contacts the region corresponding to the column. And we've seen an expanding catalog of Hi-C data in recent years. Here we see the data portal for the 4D Nucleome Consortium, which curates numerous Hi-C data sets from independent conditions, from multiple time points, as well as in disrupted or atypical cell types. And we have an opportunity to interrogate these diverse data sets to find patterns and changes at multiple structural scales. And this is something that we've also just seen on Myers' talk. When we look at a Hi-C matrix, we see compartmental patterns, heterochromatin being one of the kind of compartmental pattern we see here. And that segments the genome into a transcriptionally active, open chromatin regions, as well as the more repressed coiled up regions. And if we zoom in, we see TADs, or topologically associating domains, which are these groups of neighboring regions that tend to interact a lot with each other. And then in the closest, most granular scale, we have chromatin loops, some of which facilitate that enhancer promoter contact, as we've seen earlier. We're in particular interested in changes at the TAD scale. And why is that? What motivates that kind of interest is how TAD level changes can lead to diseases, including cancer. So here on the left, we see an enhancer in this red dot that is insulated from accessing a gene in this green box over here. And that insulation happens because they're in a different TAD. But disruption to the TAD boundary leads to this ectopic enhancer-promoter interaction in this fused TAD, leading to the activation of a particular oncogene. TAD scale changes are also observed during differentiation, which we again just saw. Shown here is the Hi-C data from mouse neural development time course, starting from mouse embryonic stem cell, going to differentiating all the way to a cortical neuron. We see a TAD split right along this middle here, and there's corresponding gene expression changes, particularly for this ZFP608 gene that is in the vicinity of that particular TAD split. In the reverse process of pluripotency reprogramming, so here we're going from a differentiated B cell back to a pluripotent stem cell state, we also observe this kind of TAD scale changes as well. And finally, TAD dynamics may also be a factor in evolution. So here we have a particular type of retrotransposon site marked with this arrow right here. And those sites tend to demarcate a TAD boundary when they're transcriptionally active, but only in great apes or human and great apes stem cells. And as the evolutionary distance grows, that particular element no longer appears to facilitate that TAD boundary enforcing role. So you can see that in New World monkeys and non-primates down here. So while we have many anecdotal evidence that TAD scale structural changes have downstream consequences, it's still an open question when we look at genome-wide data, when exactly it plays a significant role in changing gene expression. So here we have two recent publications, one demonstrating that changes at TAD level doesn't really seem to affect the majority of gene expression levels, or only 14% of the boundary deletions resulted in change in expression in nearby genes. So we need an effective way to narrow down and pinpoint regions that are involved at TAD scale in order to link structural dynamics to downstream consequences, especially when we have genome-wide data sets like those generated from Hi-C. So if we distill this aim down to a computational task, we have as input multiple input Hi-C data sets from a related biological condition or from a biological process like differentiation. And we want to be able to provide our collaborators, biologists, lab scientists, with an interpretable output that annotates each genomic region with context-specific boundariness. So here in an example output bed file, each row represents a genomic region, and it has an annotation about whether that particular genomic region has a static shared boundary or some kind of a context-specific boundary. So we developed a pipeline that involves a multitask factorization framework and some downstream significant testing and annotation, which we will walk through now. So the core component of our proposed method is non-negative matrix factorization, or NMF, and this is relevant. The non-negativity is relevant because high CA matrices in most normalization schemes are non-negative. Just as you can, so what is NMF? So just as you can factor a large number, or any number, really into two smaller numbers, most numbers, you can factor a large matrix into two smaller matrices. And these smaller factor matrices can be used to co-cluster the row entities to the column entities. So if we consider the data set, this matrix, matrix from the Netflix challenge, where we have the user movie ratings, these factors can help us identify, in this particular instance, two broad groups of users that are interested in two broad genres or groups of movies. Formally, the objective of NMF is to find these two factors, U and B, that when they're multiplied together will approximate the original matrix X. So we extended this to a multitask framework called TGIF, which stands for Tree-Guided Integrated Factorization, so we can naturally compare across multiple contexts. So in this multitask framework, each task corresponds to a biological condition or a cellular state and has its own input high C matrix. And then for each task T, our goal is to find factors U and V that will approximate X when they're multiplied together, so just like in a single task NMF. And the factors from tasks that are closely related in the task tree will be constrained to be more similar to their shared parent factor. This constraint is encapsulated in this regularization term in the objective. The tree itself is specified by the user. It encodes any prior knowledge about the task relationship, allows us to handle data sets with arbitrary relatedness, and also flexibly capture task heterogeneity. And to learn these factors that optimize the objective, we use a type of block coordinate descent algorithm with a guarantee to converge to a local minimum. Now, once we have the factors, what do we do with it? We could treat the factors as latent features representing the genomic regions and then cluster them. This is what a lot of people do with NMF. We could then identify regions that change cluster assignments from one context or task to another. So here we have a stretch of regions that went from the orange cluster to the blue cluster when they're going from task A to task B. Unfortunately, this approach had high false positive rates in identifying these dynamic regions in simulated data, especially as the number of tasks increased beyond 2. So instead, adopted a downstream processing approach similar to TAD compare, a method that we'll benchmark to. We calculate a boundary score for each genomic region. And this boundary score is essentially the cosine distance between a given region and its immediate upstream neighbor in this lower dimensional space captured by the factor U. And the idea is that this boundary score should reflect changes in the interaction pattern back in the original high dimensional high C matrix. And since we don't know how many TADs or clusters of interacting genomic regions are in a given chromosome, we sweep this hyperparameter K, which is the smaller dimension of factors, for each of the submatrices we are applying TGIF to down the diagonal of the high input high C matrices. And then we take the mean boundary score across these Ks for each genomic region. So this is one of the intermediate outputs of our pipeline. Now we're almost to the end. To provide a more interpretable output with a list of meaningful significant boundaries, we want to be able to flag those significant boundaries from the boundary score. So we compare the mean boundary scores for each of the regions against the distribution of background signals we generated separately. If you want to know how this was generated, please come talk to me afterwards. And having this kind of null distribution background allows us to calculate an empirical p-value of all of our boundary scores for each of our genomic regions. And once we have p-values, we can do significant testing and any multiple test correction. So once we have identified the significant boundaries, then we can finally annotate each region for their context-specific boundariness or their persistence in domain boundaries. So we have our final output bed file where each row corresponds to a genomic region or bin, and we have some annotation about whether a shared, static, or context-specific boundary is found within that region. We benchmark TGIF to two existing approaches for identifying differential boundaries. First approach is to use robust independent TAD calling method and just manually find differences in boundaries in two tasks. The second approach is an existing differential TAD finding method that I mentioned briefly earlier. We generated simulated data sets with known boundaries following the procedure from Furcato et al. And then we measured precision, recall, and F-score against the ground truth known boundaries. And we did this for different types of boundaries, so task-specific boundaries, as well as static shared boundaries, as well as cross how distinctive the two input TAD sets were in the simulated data, so, which is measured by this number of differential boundaries. And we find that TGIF has the highest precision across all those settings, both in task-specific boundaries, shared boundaries, and across different degrees of task distinctiveness. We see that there's a slight decrease in recall compared to other methods in all but one comparison task. And overall, TGIF has the highest F-score in recovering differential as well as shared or static boundaries in simulated data. We next applied TGIF to cardiomyocyte differentiation data with high-C matrices captured, high-C data captured across 6 different time points. Here's an overview of the number of boundaries identified by TGIF. Around 35,000 boundaries are found in the union of boundaries across all time points, and there's about a third of them are found in the intersection of boundaries from all of the different time points. And then within each time point, we have identified about 20 to 23,000 boundaries across all chromosomes, and then about a tenth of them are only found uniquely within each time point. Zhang et al., who originally generated this data, and they originally used this data set to show that a particular type of retrotransposon called ERV-H, when they're transcriptionally active, demarcates TAD boundaries in human pluripotent cells. So you see here at day 0, in stem cells before differentiation, a transcriptionally active TAD boundary, which is marked by this vertical line here, it forms a strong TAD boundary, which dilutes out as differentiation proceeds. So validating this point, when we look at the top 100 transcriptionally active ERV-H sites out of several thousand, we find higher TGIF boundary scores at the day 0 pluripotent state than in any other subsequent time points during differentiation. And here we show as an example one such top transcriptionally active ERV-H site, which is marked by this gray vertical dotted line that coincides with the significant TGIF boundary, and that disappears in subsequent time points. I'm only showing the first 3 time points here, but in the subsequent time points, we also observe a similar pattern. Finally, we've started to analyze disease-associated GWAS SNPs and their relationship with differential or static boundaries that are identified by TGIF. Among about 4,800 SNPs, 4,800 SNPs associated with cardiovascular disease in the GWAS catalog, about 10% were found in a boundary in at least one of the time points during cardiomyocyte differentiation. So here is an example, we're showing 2 GWAS SNPs and their associated genes that are falling on a shared TGIF boundary or a static TGIF boundary marked by this gray vertical line. And we plan to further analyze these SNPs for different types of enrichment and boundaries, different types of boundaries, and demonstrate how TGIF can be used as a hypothesis generation tool in understanding variation. So in conclusion, TGIF is a multitask matrix factorization method to identify regions involved in dynamic 3D genome organization. It effectively recovers ground truth TAD boundaries and simulated data, and we applied it to cardiomyocyte differentiation data to show that we can flag context-specific boundary elements, and also we hope to show down the road how it can be used to find link structure to variations that is associated with a disease. I'd like to thank my advisor, Sushmita, and the members of the Roy Lab. We're able to do our research thanks to the funding from the NIH and the CIB and training program, as well as the computing infrastructure that's maintained by the Center for High Throughput Computing. Thank you for listening.
That was great. That was great. Thank you.
Thank you.
Time for questions.
Should I exit out or? I'm okay. Okay.
Thanks, that was really cool.
Cool. Thank you.
I like the model and I'm curious for a little more detail.
Who's asking the question?
Um,
Oh, I'm sorry. Go ahead.
One, I guess two somewhat related questions.
Mhm.
For the tree structure,
Yeah.
one thing is, do you give it as input or is it inferred? And if so, how is it inferred? And then the second thing is, in your loss function, you have a second term, which is the similarity along the branches of the tree. And I'm curious what kind of similarity you're using there.
Great question. So for the first question, it is user specified. But we have found that even if you're not exactly sure about the structure of the tree, if you just give it as a flat tree where everything's connected together, it is still pretty good about picking out the differences that you see, at least in the simulated data. And we probably try to suggest, just based on high-level correlation of your high-C matrices, how you can build one if you're not quite sure. But it's really just a way to facilitate any prior knowledge that you have about the relationship between your input data set. For the second one, so all it is literally you are we're trying to calculate the Frobenius norm. So the difference between that, it's like the closest thing is Euclidean distance of vectors, right? Except, it's the Frobenius norm. The differences between the two, the child factor and the parent factor, and that's the thing we're penalizing if it's too large. Hopefully that answered your question.
Yes.
Awesome, thank you.
Great, Doctor. I really like it.
Thank you.
I have a question regarding the SNPs. Did you try to look whether they affect any regulatory mechanisms like binding sites for CTCF or other reorganizing factors?
That would be our dream. I mean, the idea would be that maybe the SNP itself is in fact disturbing some kind of a binding site of either context-specific boundary element or more static, something like CTCF. One thing that is not included in here, but the analysis I just included is looking for motif sites that are specifically enriched across all these boundaries and as well as like in the unique boundaries as well. So hopefully we can try to bridge those two analysis. Yeah.
Thank you.
Thank you.
Thanks. It was a great talk.
Thank you.
I was wondering if you take into account the tree structure when you are defining, I think you are not, but whether you consider taking into account tree structure when you're calculating the differentiality, because I believe the leaves of the tree will be if they come from the same parent, they will share more similarities. And do you actually see like less distinction if the cell types, for instance, are more closely related to each other in comparison to the other?
Yes, so this is something that I tried in like our early experimentation. But yeah, if we just basically measure the correlation between the output factors themselves as well as the Jacquard similarity between the clusters that we get out if we do not cluster on the factors, it does basically obey the structure of the tree. So factors coming out of the leaves that are sharing the same parent, they'll be more similar. And you can increase and crank up the strength of the regularization, then you see more things becoming more and more similar. So that's something that we'll have to just suggest how to like set that level of kind of enforcement of the similarity. But yes, it does tend, the output does tend to obey what we're telling it.
And apparently your differential boundary are the ones that are unique to the given condition.
Yes, we are actually trying to figure out all the right words to call these different type of boundaries, because when I say something specific, it means it's found at a time point, but should I also mean that it's unique in that boundary? Because it could be found, a boundary can be found across multiple time points. So if you have any suggestions of all the different options of words that I have, they're specific, unique, all that, I'm open to suggestions.
That's hard to do syntax. Absolutely, good luck with that.
Thank you.
Just to extend out on the questions about the tree topology itself, are you limited to the tree topology, or is there something about it that's better in your setting? If you knew your data were linearly, across a differentiation time course or something like that, can you have an arbitrary structured graph?
You could. It would be, we started with this tree structure because it's the easiest way to attack something like a cell lineage or like cancer subtype data sets. A lot of data sets that we're interested in is also linear, like you said. In that particular case, you could just hook up neighboring, tasks, and then in each task that is not at the end would have two parents. So the algorithm and the object could be easily adapted to handle that. But it's slightly more computationally expensive because now we have to look at two different parents instead of just one for each of the leaf nodes. So the extension is absolutely possible. We haven't looked at it yet.
Okay.
Thank
Okay.
you.
Thank you.